Hi, I'm Brad Power, and this is the Cancer Patient Lab. And let me just cover our standard couple of housekeeping items before we get started. The first is that this is not medical advice. This is for information purposes only. Everything that you hear is with the goal of allowing you to take some of these ideas to your medical team for consideration in your care. Second is, I call it the Miranda rights. Everything you say can and will be made public. So if you're concerned about that, hide your image, change your name, and don't say anything. And finally, we are a patient-led learning community. We would appreciate any donations that you might make, which you can do through our website and the donate button. Just to make the connection, maybe Brian, since you're here, maybe you can introduce Dr. Gross just quickly and describe how we got connected and your background with him.
Sure. Yeah. So actually, our paths go back many years. So I was diagnosed with prostate cancer in 2016, and Dr. Gross was part of my clinical team very early on, I think as early as 2017. And he's been part of my care team really ever since. But I got interested in repurposing drugs and reached out to a company called RebootRx. I don't think Laura Kleiman is on this call. But long story short, I put Laura in touch with Dr. Gross because he is an expert in the repurposing of drugs. And Laura had a very interesting AI methodology for identifying drugs that could be repurposed, but of course it requires clinical scrutiny. So I don't think that we're going to be talking about that so much, but we are going to be talking about antidepressants and the amazing research that Dr. Gross has done with antidepressants in combating prostate cancer and other cancers as well. So Dr. Gross, thank you so much for accepting the offer for to be here, and we look forward to your lecture and very active discussion from the community as well.
No, that's great. Yeah, thank you again so much, Brian and Brad, for inviting me to speak. So let me hopefully share the right slides here, and we can start. There we go. Okay. Is everything, can you see everything okay?
You probably want to go to, there you go. Now you're getting there.
Okay, there we go. All right. So again, thank you so much. So really happy to present. So what I thought I would do here was kind of talk about the story of monoamine oxidase inhibitors, and how I propose that's related to prostate and other cancers, and show you really some data, give you a feel for the data in prior publications that sort of led me to this conclusion. And then at the end, we'll talk about some next steps and some opportunities that I see. But
Dr. Gross, the way this is showing up is it shows your slide and then the next slide and notes. And so you probably want to get on presentation view or something like that. So it's just the main slide you're using.
Oh, okay.
Anybody know their way around PowerPoint who can switch it to presentation view? There you go. No.
Yeah, I don't know why it's yeah, I'm just having
or monitor at the top right beside the cancer lab with the down arrow, and then you'll see a monitor. Press there. Start from the beginning.
Yeah. Oh, it's giving me
yes.
It's finding the wrong screen.
Yes.
I think if I do, is that better or no?
No.
Now,
no.
once you get in there, if you click the display settings at the top.
How about that? No.
Oh,
Not good.
Yeah, see display settings?
Yes, hit the display settings at the top in the middle of your screen. It says show taskbar, display settings, and end slideshow. So go to display settings. Next one over.
To the right.
Right.
Move your mouse to the right.
Okay.
Uh-oh.
I think I have something on the middle.
Yes, it
There we go.
says show taskbar at display settings and end slideshow. At the display settings at the top of your screen, you're almost there. Okay, that show taskbar, go to the right. A little bit more with your cursor to the right. To display settings, move your mouse to the right.
Yeah, I think what he's seeing and what we're seeing is very different.
Yes.
Yeah,
Are you
it's
seeing,
different.
are you seeing the PowerPoint screen, the PowerPoint menu?
Yeah.
If you are, go to view and set it into presentation mode.
So yeah, you're not seeing this. Okay.
If you see the PowerPoint, it would be under review or view, probably view.
On the bottom, the thing that looks like a screen.
There you go. There you go.
Yeah.
Into well, let's see.
Yeah. Do you know what?
Oh, go to Slide Master. Go to slide.
No.
I mean, you're in the right spot
You know
now.
what?
This is where
I'm having
this is
to-
where you make the changes.
The bottom right, there's a thing that looks like a screen. If you click on that screen, you'll get it.
There you go. That's
Yep.
we're moving in the right direction.
We're almost there.
This is so Brad, can you edit this out when you start? Okay.
Yeah, no, it'll get edited
go-
out.
Go
All
to
right.
fit. Go to fit to window. That might zoom fit to window up at the top. Oh, lower. Yeah, to the left. Did you
Yeah, that
did you
one.
click on zoom or fit to window? A little below that, a little down, a little to the right, a little more to the right, a little more.
Okay,
Zoom or
Zoom.
fit to window.
It's not letting me do that.
You can click on 100%
Yeah.
or fit to
Yeah.
window. Zoom would probably work. Did you click on zoom? Well,
I
Yeah.
still think
You're
it's
sort of
down on
there.
the bottom right. Down on the bottom right, there's a
Yes,
screen.
there you go.
That one. Click on
that
that.
should work.
No. You were just there.
That
That's it.
here?
That one.
Yes.
Yeah, see,
slideshow.
but it's
Yes. No?
Okay, do you know what? I'm gonna do one other. Let's see. It's a two monitor problem. Okay. Is this
There you go.
Beautiful.
okay?
Did it.
All right. Okay, thank you everyone for your patience. But okay, let's start. Okay, how did I get here? All right, so monoamine oxidase inhibitors. So first we'll talk about monoamines, and we'll talk about monoamine oxidase inhibitors, and then we'll talk about how these are related to prostate cancer and other cancers. And then we'll kind of talk about some next steps. So what are monoamines? And let me check, hopefully everyone can hear me okay. But so monoamines is a class of molecules, natural signaling molecules in the body. There are many of these that are really important for many functions, especially in the brain. So we know a lot of these in like the popular literature. There's what we call epinephrine or adrenaline, that's a monoamine. Other ones are called like serotonin or dopamine, histamine. These are all monoamines. So these are very important molecules for lots of processes in the body that has to do with signaling, basically telling one cell or another cell what to do. So some examples of these monoamines in diseases include in depression. So low serotonin levels in certain areas of the brain are related to depression, and we use drugs that try to raise those levels. In Parkinson's, the main problem in Parkinson's is a deficiency of dopamine in one particular brain area. And again, we use medicines to raise those. And then we know about in what's called, adrenaline or epinephrine or fight or flight response. So that's also driven by a monoamine, by epinephrine, which is, has to do with blood pressure and heart rate and all attention and all sorts of things. So that's kind of the function of the monoamines in the body, some examples of that. So what are monoamine oxidase inhibitors? Well, every time you have an on switch in the body, you also have an off switch. And so really the monoamine oxidase inhibitors or monoamine oxidases are kind of the off switch. So if you look on the right here, we see a kind of a diagram of the monoamine system, like two neurons talking to each other. The purple dots, the triangles represent monoamines, and they kind of go between the cells. But then on the left, the monoamine oxidases will turn off, will inhibit those. And so it helps to regulate the right amount of the neurotransmitter as they're kind of communicating to each other. So that's the picture on the right. On the far right is if you have a monoamine oxidase inhibitor, what you do is you prevent or block the inhibition or the degradation of those purple triangles. So the purple triangles build up, you get more monoamine, more purple triangles, more signaling to the other neurons. So basically that's what monoamine oxidase does. It helps to regulate this very important system in the body that has lots and lots of different aspects of it. So the other part to go into is there's what's called monoamine oxidase A and monoamine oxidase B. So these are two forms. The important thing there is they're expressed in different parts of the body, different parts of the brain, other body parts as well, but also the chemical inhibitors that we develop can be specific to one or both of these. So that's kind of, there's MAOA, monoamine oxidase A, and MAOA B, monoamine B. They have different areas, they have different functions, and then the inhibitors also have different activities, different functions. So most of the monoamine oxidase inhibitors, certainly the ones that are used somewhat in psychiatry now, but not so commonly, used to be are non-specific inhibitors. They inhibit both the A and B forms. There's another MAOA inhibitor, which is much more commonly used in Parkinson's disease called selegiline. It's either a patch or a pill used in some patients with Parkinson's. That's very specific for MAOA-B. So there's a kind of a complicated system, very important system, but something that we can basically take advantage of for cancer treatment, and that's sort of what we'll talk about. Okay. So how does this at all relate to cancer and prostate cancer? So what I'll show you is that monoamine oxidase A, especially that A isoform or the A form, is very important for prostate cancer. It's highly expressed in high-grade prostate cancer. Also, our data that I produced and others show that MAOA in particular can be modulated to decrease the development of prostate cancer in animal models, and also is a target of treatment in animal models. And that when a cancer becomes resistant, MAOA can be one of the proteins, the enzymes that's highly upregulated. So a little bit, because I know this is called the cancer lab, so I thought I'd do a little bit of sort of lab history here. So why do we think MAOA is upregulated in prostate cancer? So this is some very early research that was done now 18 years ago by Larry True at University of Washington, that basically you can take prostate cancer and there's an area that's called low-grade Gleason 3, and we know there's this kind of pattern on the lower left, or the yellow that represents the Gleason 3. And there's other areas that looks very aggressive. We call it Gleason 4, Gleason 5, and that's in the upper right of the photo micrograph there. And so what we can do is use a laser to basically cut away those two pieces of a low-grade and high-grade prostate cancer, and then try to look at using genetics and other features, what is different between those two areas. And so this was one of the first studies that did this way back 18 years ago. And when they did this, they showed that one of the most important genes, the most commonly overexpressed, the most important genes, and also the protein that was differentially expressed was MAOA. So MAOA, monoamine oxidase A, the gene that we're talking about, is very, very highly expressed in high-grade prostate cancer and in many other cancers as well. But this is especially relevant to prostate cancer. Okay, and then this is some other data about treatment. So I mentioned that MAOA is upregulated in treatment-resistant cancers, including especially prostate cancer. There are many studies that have shown this, both in patients and in animal models. This, I think, is the most compelling and one of the more recent ones. So you can grow human prostate cancer in an animal model, in a mouse called, this is enzalutamide C42. This is a model of treatment-resistant prostate cancer, enzalutamide-resistant prostate cancer. And when you treat with a MAOA inhibitor, so chlorgeline is an MAOA specific inhibitor, phenelzine is a non-specific AB inhibitor, you can get some decreased growth of the cancer. So that's very, very encouraging for sure. But even more encouraging is if you combine the chlorgeline MAOA inhibitor with enzalutamide, or phenelzine with MAOA inhibitor, phenelzine, enzalutamide, you'll get even more inhibition. So this is, I really think, the best data so far about MAOA inhibitors in a preclinical model. And there's some questions. This is so great, but maybe let me get through about 10 more slides or 9 more slides, and then we'll do questions if we could. But I'll try to monitor the chat, and maybe Brad can help me as well, make sure I don't miss anything. So monoamine oxidase inhibitors. So you know, what you know, this is everyone, or not everyone, but many people might know that this is a very old field. That I know in medical school, I learned that MAOA inhibitors were the drugs never to prescribe because they had lots of problems. And I think that's more or less maybe still true. But so monoamine oxidase inhibitors have a very long history in medicine. So the one that I've especially studied is this one called phenelzine Nardil. So Nardil was first used in patients in 1961. So very long time ago. And here on the right is an ad from a British medical journal. I think they used to allow ads. In the United States, you know, they didn't allow ads for medicines for, you know, decades. But I guess in the UK they did. And there's this woman here who's brought to the doctor by her husband because he's out of his league and he needs the psychiatrist help to diagnose menopausal depression, and Nardil is the treatment. So yeah, so times have changed, medicines have changed, but certainly this is a very old drug. So then if we go forward in the 70s, monoamine oxidase inhibitors, but antidepressants were really changed a lot. Many new antidepressants came on the scene with less side effects. We'll talk a little bit about that, but and certainly more effective. So definitely came out of favor for a treatment. But still, monoamine oxidase inhibitor, phenelzine or Nardil, is used rarely in some patients with depression. And I think there's some psychiatrists on that could comment on that, but you know, very, very rarely for something called atypical depression. Sometimes it's used, or atonic depression. And then MAOA inhibitors, as I talked about, are used much more commonly in Parkinson's disease and for neurology. But so that all of this background, you know, brought me to this trial that I was able to present and publish now 4 years ago or so that looked at monoamine oxidase inhibitor in prostate cancer. So I think still to this day, this is the only published trial showing activity of monoamine oxidase inhibitor for any cancer. In this case, we chose what's called biochemical recurrent prostate cancer. So you will know that when we call biochemical recurrent, what we mean is that imaging studies are negative, but the patient has, you know, maybe they had a prostate surgery and their PSA is 1 or 2 or 3, or they had radiation and the PSA is 2, 3, 4. So this is like the cancer's coming back after primary treatment, surgery or radiation. But we do scans, and certainly back then it was a bone scan, CT scan, that did not show any cancer, and we call that biochemical recurrent prostate cancer. And so what I did was I led a clinical trial testing phenelzine, again, this old monoamine oxidase inhibitor, really one of the oldest, but clinically available and used. What I found was that about 24% of the patients had a PSA decline. So that was definitely, encouraging. But even more, about a third of the patients had no progression. So some activity of phenelzine monoamine oxidase inhibitor in prostate cancer. But I also found, the side effects of these drugs, and certainly we were aware of that starting the trial. But about 25% of the patients discontinued for side effects. So it's this is patients without depression. We're giving an antidepressant, and there's a lot of known side effects for these drugs. So I know there was some discussion before about like what are the side effects. So I mentioned a little bit, and we can talk more. But the main things people really worry about with phenelzine or monoamine oxidase inhibitors are drug-food or drug-drug interaction. So there's this very important drug-food interaction that tyramine in the diet. So tyramine is an amino acid that is part of the diet. If that goes into the bloodstream unaffected, you'll get a very high level of tyramine, and then epinephrine. The tyramine is the precursor for epinephrine, and that could cause high blood pressure, or wildly high and low blood pressures, cause a lot of problems, and in very rare cases could be even serious and life-threatening. So for patients on the trial, we had a lot of education about diets that were low in tyramine. So tyramine is part of certain diets, especially fermented foods. So it's funny, in the old days, we would tell patients about aged cheese and red wine, but I'll tell you, in our trial, the patient that had the biggest problem with tyramine was overaged avocado and Mexican food. So back in the 60s and 70s, there probably wasn't a lot of aged avocados that was being eaten, but I think in our day we would have to tell them much more about avocados. Anyway, so that's the drug-drug food interaction problem. Generally manageable with careful education, and then drug-drug interaction. So certain drugs are not compatible with phenelzine, especially certain blood pressure drugs, and drugs like be very dangerous would be like stimulants like Adderall and those kinds of things. So there's a lot of things to worry about with phenelzine and MAOA inhibitors in general. But still, we did see some activity. So that's sort of the prostate cancer story, but what about other cancers? It's very important. I mean, it's been shown, but in lung, breast, colon, even glioblastoma, brain cancers as well. We'll show you some data on colorectal melanoma and also Hodgkin's lymphoma. So lots of things. So again, this cancer lab, so I thought I'd give you just a flavor of some of the research. So this is more recent data. This is from 2021, a group, Lily Yang at UCLA. So, we can grow basically mouse melanoma in a mouse, and we can treat it. So the black line represents the cancer growing in the mouse very fast. We can use phenelzine, again, the same drug that we talked about, that's the red line, that will decrease the growth of the cancer. So again, by itself, it's encouraging that it can decrease the growth. But more than that, there's this anti-PD-1, which is the blue line, which is kind of like this drug pembrolizumab or Keytruda. This is something called the immune checkpoint inhibitor that stimulates the immune system. And so what this research showed was that phenelzine, so the green line, phenelzine plus this kind of checkpoint inhibitor really accelerated the anti-cancer effects. So this is a very striking result. They showed this also in colon cancer, I think also in a breast cancer model as well. So very interesting that phenelzine or monoamine oxidase inhibitors can help basically promote the immune system to recognize cancer, especially in the context of checkpoint inhibitors. And then this is, I think, some of the most striking data, but also really some of the oldest data. So procarbazine is a very old cancer drug. In fact, it's really revolutionized the whole theory, the whole field of oncology. So back in the day, the big question of oncology was, could we use chemotherapy to treat or even cure cancers? And the representative example of that was Hodgkin's lymphoma. So Hodgkin's lymphoma, very still rare but important kind of lymphoma. Back in the day at the National Cancer Institute in Washington, D.C. and in Maryland, they were testing different kinds of chemotherapy to try to treat this really aggressive lymphoma. And their regimen, which was a combination, the idea of combining medicines, was very limited because of side effects. So they, on the left, this was the initial data they had with a regimen called MOMP, M-O-M-P. Those are the abbreviations of the medicines that comprise the regimen, but it was not very effective. It worked a little bit, was not very effective. But really tremendously, what happened was when they added procarbazine to the MOP, you saw tremendous activity in anti-cancer activity. In fact, other data showed you cured the lymphoma patients a good percentage of the time by adding procarbazine, which is this drug. So procarbazine was developed as a monoamine oxidase inhibitor. Later, we found it to have other anti-cancer effects, but overwhelmingly, this kind of data showed that maybe combining it could have activity. Okay, let's see. So that was kind of where I've been, but where I'm going is trying to develop new medicines that really design out the bad side effects of the monoamine oxidase inhibitors. So this data we published just about a year ago now, working with my colleagues at USC. We basically are redesigning the monoamine oxidase inhibitor scaffold, we call it, and trying to get it not to go to the brain and only to focus on the peripheral activity, which is where, you know, certainly as a prostate oncologist, we'd like it to stay. So this is, we basically designed some new medicines. We, you know, have it, you know, submitted for a patent, and we're trying to further develop this project to look at monoamine oxidase inhibitors, basically redesigned, that don't go to the brain. So that's really my summary. I know Brad wanted me to only kind of give half an hour. I could always talk more, but monoamine oxidase inhibitors, there are many forms in cancers. Monoamine oxidase A especially is related to cancer. We can use inhibitors against the monoamine oxidase inhibitors, and I mentioned some of them that are used. Chlorgelline is only available in research, though there were trials about maybe 20, 30 years ago that did test it in patients, but now it's only made for research. Other MAOA inhibitors are available, phenelzine being the one that we talked about, which is a non-specific AB inhibitor. But people are developing new versions, and especially our group are hoping to develop one specifically looking at the peripheral activity. And always we're looking for additional support data, you know, volunteer, to help us really understand how this could be used for cancer treatment. So that's it. So that was sort of the prepared talk. Let me, if I could, I'll get out, and I can maybe field any questions if there are.
Yeah,
There were a lot of questions in the chat.
yeah, I was going to say,
Yeah.
Alan has some questions. They're mostly from Alan in the chat.
Okay.
Alan, you've got at least three. Maybe you could start with one to try and sequence through them.
Okay. I'm very long-winded. I put a lot of people off. I thank Brad for not ejecting me from this cancer lab. In the chat area, I gave short, quick questions. I guess I could read them, but I actually have some big honker questions that are compound.
Sure. Okay. I'm going to try to monitor the chat, but go ahead. Why don't, if you could summarize it, that would be
You
great.
want me to read them, or can you read them?
It would be easier if you read them for me.
Okay, so I
Technologically,
dragged my wife along, Michelle Sager, who's a psychiatrist, and so I just asked a basic question. One of the problems with any kind of phase 1, phase 2 is not efficacy, but safety, hearkening to do no harm, Hippocrates. It turns out psychiatrists have as you showed historically, have 50 years of experience giving these drugs. And there's a reason they call things first generation, second generation, and third generation, is because the first generation weren't as good as the second generation. So, so I'll just add, it's simpler for you just to read my question because it's shorter. But I presume you do not have long-term experience. What is your, are you a urologist? I didn't get that part of it. What are you?
I'm a medical oncologist.
You're a and my belief is, but you're going to correct me if I'm wrong, most medical oncologists at university centers are subspecialized in a particular organ system. Is that correct?
Lots of academic centers, yes. And I am a prostate cancer oncologist.
Okay, good.
I do general urinary and prostate.
So we know who we're, that's another soapbox I have about cancer patient lab. Since we're a Tower of Babel, everybody should know first and foremost what your area of expertise is. You are a medical oncologist, and furthermore, you're subspecialized in prostate cancer. And therefore, I'm going to say something that might sound pejorative, but is not, which is you do not have long-term experience giving MAO inhibitors. Is that correct?
Are you involved in any litigation? Let me ask you.
I actually am, but it's on the receiving end. But that's okay.
Okay. No, but so if the question, if I could answer it in the general sense, so monoamine oxidase inhibitors have been used long term in psychiatry for many patients, and there are patients that have been MAOA inhibitors for years and years and decades. So so I think the safety of these medicines, I mean, that's one of the advantages generally of the repurposing strategy, is that we know a lot more about the safety, we know a lot more about the toxicity, but there are always uncertain questions. So overall, MAOA inhibitors in animal studies and in human studies have been very safe in the long term. But the ones that have been developed...
I looked at your study, you had 24 patients and you had a grade 4 hypertensive episode. So that,
right.
In my math, says that's 4%. And admittedly, there's a big plus or minus because your denominator is 25.
Yeah. Yeah. No, I mean, as an oncologist treating cancer patients, we take this very seriously. And certainly the adverse events, any one is terrible, and we try to avoid them. We are trying to balance it against the benefit of treating the cancer. So yeah, this is why, though, my study was preliminary pilot. It was, I think, encouraging, but I didn't propose, and nor would I, that thousands of patients should start this phenelzine. You definitely have to balance the risk and the benefit. Any physician will know that.
That actually segs exactly into what I thought was very tangential, my second question, which appears completely different, but is not. So if you could read it, or do you want me to read it out? You can read it.
I have trouble navigating the
okay,
the
I'm going to tell you. Here's I'm going to read it to you.
Yeah.
I believe it's connected, even though some people might view it as loose. Your study is from 2020. Do you know how many clinicians are using? You already answered this.
Yes. Okay. Yeah. So the question of I mean, I don't think there's widespread uptake of phenelzine in cancer. No.
Why not?
That's
Why
true.
not? We have this off-label.
I don't know. It's a good question. Yeah. I don't know. I mean, I think there's most certainly data to be explored. You'll have to ask the community why. I could tell you, as a practicing physician, at times I've given it to patients, but you really have to think a lot about, as any physician does, the risks and the benefits. So in cases where we have a better option, or more safe or more well-proven option, we would choose that option. And I think any good physician would do that.
Yeah, I really got to ask you this question because I'm stupid about this, because I'm a pathologist and we're the field that absolutely is the least familiar with therapeutics. I do not treat patients. Unbelievably, I'm a doctor, but I don't treat patients. What is your definition of off-label? Because I still to this day do not understand it.
Okay, so off-label really means basically prescribing a medicine outside of the FDA-approved label. So the FDA-approved label for a medicine like phenelzine would say it's used for antidepressant. There's some maybe benefit in Parkinson's. It's a little bit complicated because the ones that were prior to the current FDA regimen, meaning the current laws are sort of grandfathered in. So there's a kind of I think a little different loophole for that. But yeah, so technically that's what the label use. So if you're using it for the indication that's not the, the FDA approved indication, you can also adjust the doses as well. And so sometimes off-label means giving the medicine for the same reason, but the doses are higher. So those are a couple examples of
Okay,
Alan,
Where's
let me
Alan?
interrupt so we can give some other people some airtime.
Sure.
You can come
Yeah.
back if we have time at the end. Brian, you have your hand up.
Yeah, so Dr. Gross,
Mhm.
do we have any data that would suggest what type of patients would benefit most and least from this type of drug? We have so much data now. We have genomic, transcriptomic data. It's proliferating. And is there a way for us to understand, which of us would benefit and which ones wouldn't?
Yeah, so that's another sort of pillar of my research, and I don't have much, public to say about that, but looking at, genomics in response to treatment, and maybe that would give us some data. In terms of the published data, so there were studies that looked at prostate cancer treated with chemotherapy, and this is what's called the neoadjuvant study. So basically give a patient chemotherapy, and then they would do a surgery, and they asked the question, what genes changed in the cancers, especially the ones that were resistant to the chemotherapy. So like you take a sample before treatment, and then you give treatment, and then they get a surgery, and then kind of compare those two samples. MAOA was upregulated in those patients. Now, that's it's not everyone, and this is one of the challenges. It's really, is it everyone? Is it most? It's one of the most commonly upregulated treatment chemo-resistant, Taxotere-resistant genes. And that, in fact, there was a trial that was started but then didn't finish at University of Washington and Oregon that looked at giving Taxotere with phenelzine. So yeah, so we don't really know. It seems to be very upregulated in treatment-resistant prostate cancer, and in other cancers as well. Trying to identify that prospectively, I don't know. It is a gene that you can, look at in gene expression arrays, and the protein you can look at. But yeah, so this is one of the big challenges, because it at least in animal models and in humans, it does work in some, but not all patients.
Got it. Thank you.
Dr. Sager, you're up next.
Hi, thank you. Dr. Gross, thank you for such an interesting talk. And you're at USC, is that correct?
I'm at USC and the Ellison Institute here in Los Angeles.
Oh, wonderful. Yeah, I went to medical school there, so I'm familiar with those programs.
Great.
I'm curious about how you're trying to adjust this MAOI so that it's not, so it's an MAOI-A, but it's not going to cross the blood-brain barrier. So, I'm curious about this. So the idea is that it's some sort of putative immune modulator somehow is activating.
Well, it has a direct anti-cancer effect.
It has do you know, I guess I have two questions then. What is your notion of its mechanism, number one? And two, what is it that you're trying to do by keeping it from going into the brain? Because I'm wondering if it also has some sort of possible beneficial effect there also.
Yeah. So the mechanism of MAOA in the peripheral cancer tissues is one question. And then the second one is maybe this is, maybe it's not an off-target effect, maybe it's a desirable effect to get it to the brain. Those are two very important questions I can speak to. So the first one, this issue of how does MAOA work in, I showed you the picture in the neurotransmitters in the brain, but how does that have to do with a cancer cell in the prostate or, lymphoma or some other cell? So MAOA is, it has an intrinsic function inside cells if this means anything, it's actually a mitochondrial resident enzyme, and it regulates the redox potential inside the cells. So monoamine oxidase has a more, we call it pleiotropic, a wider effect in tissues, independent of the effect on modulating the neurotransmitters. That's how it was discovered, but the actual effects are much more broad in tissues. So there have been studies, and there's a couple published studies. One, these are very prominent studies. JCI is a very Journal of Clinical Investigations, kind of very prominent journal, published about now 5 or 8 years ago, that showed the mechanism, or at least some of the mechanisms. It has to do with, again, with redox. It has to do with transcriptional regulation, VEGF, and other pathways. So a variety of pathways that are affected, but it's probably more the overall the redox potential inside the cell, which is signaling the basically cancer cells to either differentiate or not differentiate, and you want them to be differentiated so they're responsive to treatment, basically. That's so. Now the second question is there do we want it to go to the brain? So as I said, there is overwhelming data that you can take cancer cells in the laboratory, like grow them in the tissue. There's no brains involved, and we can give MAOA inhibitors many different cell lines. You'll have an anti-cancer effect. Same in animals and other things. So there is an intrinsic cancer effect, but maybe, this is really, really interesting, maybe there's an effect on the nervous system in the brain, and maybe we want to take advantage of that. Yeah, again, we don't really know. The idea here was, decades of research have tried to engineer monoamine oxidase inhibitors to only go to the brain, right? Because if I was a psychiatrist developing a drug, I would give the opposite talk, and I'd say, look, we just want to go brain. We don't want it to go anyplace else. So basically, we're kind of re-engineering that for our purposes. I don't know if that answers the question, but you are right, and it's very interesting. The immune system and the nervous system interaction is super important, and it could be that monoamine oxidase inhibitors are affecting the nerves. And there's a whole literature on peripheral nerve innervation to prostate and cancers, and maybe we're affecting the nerves, the peripheral nerves or the central nerves. We don't really know, but yeah, I mean, there's a lot more to learn.
If you had a drug that was able to separate out those two effects, then you could kind of delineate and make that distinction and try to understand it better. Yeah. Okay, I get it. Thank you.
And that's one of the things we'd like to kind of hope to do with our research, is try to interrogate that kind of difference. Mm-hmm.
We have 3 questions in the chat. Let me tee up Cheryl Middleton. Cheryl, can you ask your question?
Try to monitor.
Yeah, I'll see if I can. Well, it was a question about whether this is for prostate.
Oh, is it recurring prostate cancer? Oh, okay. So specific to a patient. So I hesitate to sort of answer that, but yeah. So I mean, the research is there in the public that we showed in recurrent prostate cancer. Many of the patients did have Gleason, 789. We did show some activity in this particular case of biochemical recurrent, but plus minus, it's something I think to talk about to your doctor.
Okay. The next question is from Richard Anders. Richard?
Yes. Thank you. That was really interesting. One quick follow-up to Michelle's question. It seemed to me, just in listening, that a lot of the side effects were probably not in the brain, but sort of in adrenal and other systems that were outside the brain. So so probably inhibiting the blood-brain barrier interaction would still not eliminate those side effects if that was the purpose.
Yeah. So I glanced over that, but let me discuss that. So again, the big side effect that we talked about here is this drug-food interaction. So this has been really well studied going back many years. So it turns out that the lining of the gut has both MAOA and MAOAB. They have both enzymes, because an important purpose of the gut is to basically remove this tyramine, the free tyramine, from your diet, which is in fermented foods and meats and things, to prevent it from going directly in your bloodstream, because that can cause this problem. So the other way around this problem of the tyramine surge with diet is if you could only, if you leave if you leave one of them untouched. So if you have an MAOA specific inhibitor, the MAOA-B, which lines the gut, is enough to deactivate the tyramine. And so the other part of the strategy, which I didn't focus on, is to do it just an MAOA inhibitor. And that way you would have less of this toxicity. So again, there is a literature in this in the I think 70s in psychiatry. There was a class of drugs called REMAs that were reversible MAOA specific inhibitors that basically avoided a lot of these side effects of the drug food interaction.
That's very helpful. That was actually not my question, but that was very helpful.
Yeah.
It was just a follow-up.
Yeah. So
my question is,
Mhm.
my question was just, you know, I used to teach life science, and you know, I remember a professor coming in at MIT who did a large study through the Medicare database. I mean, there are these enormous databases of information, and if MAOIs are not contraindicated for a wide variety of cancers, you could do sort of historical studies, I imagine, against large databases and medical records and begin to tease out interactions. Has anyone done that?
Yeah, we're as always looking for support for that kind of research, financial. But yeah, so yeah, we have talked about that. I've seen some unpublished real world evidence data from other countries. Yeah, so I think that's an enticing way to do it. One of the problems is that monoamine oxidase inhibitors, as we said, they're not very commonly used here in the United States. There are some countries where they're used more commonly because they're cheap. Like outside the United States, in Taiwan, for example, and other countries. So yeah, you could do that, and I've always wanted to do that for many years, but I just haven't had the right combination of funding.
But the drugs are not contraindicated for most.
They're not contraindicated, not at all, not at all.
Well, that would be an interesting study. I don't know how many data points you'd have. And in some, I mean, Taiwan would probably have a good registry, but you know, a lot of countries that use a cheap drug might actually not have very good records.
Right. Yeah, it's a practical problem.
Well, let me riff on that quickly before we go to Rochelle, and that is, you're doing a repurposing of an existing drug. Can you talk a little bit about what that's been like, almost in the general?
Yeah.
You have safety sort of presumably covered, or you know the side effects and how to mitigate them, and now you're trying to find a new use case or a new application for that same drug. How's that been?
Yeah. So and Brian can talk about this. And one of again, he mentioned this company called Reboot, which is a nonprofit that really their idea is to sort of mine the medical databases to kind of find opportunities to repurpose drugs. But there's a lot of headwinds. The biggest headwind is that we take a drug that has been used for one thing, and then we're trying to use it for something else, even though the physicians or the scientific community think it's safe. We have trouble. One of the problems is paying for it. So, you know, luckily these off-label drugs are very cheap, so that's a great opportunity. But no one would really want to pay for the research, because if there's no patent protection, the sort of systems being what they are, they generally don't fund the research, right? There's no drug company that will fund the research. So so we have to find other mechanisms, like either philanthropic mechanisms, angel funding, and ideas for government funding or consortiums that could help fund this kind of research. So there's this very practical thing, like it seems like a no-brain kind of question. You have a drug, you know, like, why don't we use it for something else, and maybe it can work, but getting it through the regulatory system can be difficult. There are certain, you know, opportunities and barriers. Some drugs can only be manufactured in certain ways, so maybe that's a kind of an opportunity that could be limited. The FDA does have a sort of a repurposing loophole that can give limited patent protections for certain, you know, reformulations. So yeah, there's a big practical challenges to actually making this a reality.
Brad, maybe one of the things we could do is bring Laura in, and
Yeah.
Dr. Gross. I think, you know, you could work in tandem with her on this, but she can kind of give an update regarding a couple of drugs that they are actually putting through to the NCCN guidelines. And so
Right.
there's
And she has great.
a lot of evidence to support this. And so hopefully we're going to find out soon. She may not want to talk about it until it's done, but it's in progress right now.
And she has ideas about how to get like consortiums involved that could kind of help to fund this kind of research. It's a very practical problem.
Yeah.
Thank you. Rochelle, you had some nice comments about connecting this with food.
Comments here about
Yes. So
nutrition.
in doing experimental and off-brand use with most of the patient populations that I have had worked with in the past, and as well with specifically with the pediatric population, I noticed that there would always be a micronutrient or some other food additive that we would have within the healthy components of the food, which would actually become problematic. And so I wanted to hear if there are some crosswalks that we have now that we know what these drugs have done from first and second generations, how they affect us with the micronutrients now, and how that plays a part in targeted therapies, and maybe genomic therapies. For FGFR drugs, the main issue is phosphorus. That is problematic. It breaks bones. It takes away neuropathic pathways. It unsheaths the myelin sheaths on the neurons. And so some of these become problematic for just overall survivorship. And do you know what they are for these types of drugs? And how can we use foods to either avoid but still be healthy, or is it the plant compositions that we're talking about?
Yeah, so I mean, I guess, are you thinking more about if the MAOA as a class of drugs were used, are there micronutrients that we should be aware of that may or may not influence them? Yeah, I think those are definitely great questions. It turns out monoamine oxidase inhibitors need flavines to work. It's a flavine specific drug enzyme. Yeah, I don't know. I mean, I'm sure there's a lot more research that could be done in the B12 interaction with these agents, but it's something to look into.
Happy to help and happy to look at that if you have a study or want to.
Just about out of time, but Alan, do you have one last quick question?
So I saw your mouse model studies. I think that's what those little cubes were the tumor size, and this tumor size was.
Yeah, it was published by Chang in University of Rochester.
And you showed in a sense an obvious experiment, which is to try it with the tried and true like enzalutamide. And when you combine the two, there was a greater effect. So you could use the term synergistic. Is that correct? Between the two?
Yeah.
Okay. I have this general theory, and I would like to run it by you because you are an expert physician. And it's a general crazy theory about immunology. And the theory is, I believe the female gender state is an immune better state for, I don't have a good term for it. And I base that on these observations. Observation number one, women have much greater autoimmune disease than males, substantially more. Observation number two, men get, you might disagree, you're an oncologist, you know the numbers maybe better than I do. Men get substantially more cancer than women do. And furthermore, women live longer. And then most recently, and of course this was politicized, so you could definitely disagree with this, women fared better during the COVID epidemic than men did. All these four things I'm listing for my theory that women are a better immune state. Well, if you seg that to drugs, there's a tried and true drug in your field, and it's the drugs that aim at testosterone mitigation. You know, generically ADT drugs or whatever you want to call them. Would you not? Here's the question, and I think the answer is obvious. Would you not, if somebody's going to do it off-label, it's going to go rogue. I'm going to call it rogue. If somebody's going to go off-label and use your MAOs treating BCR patients, wouldn't you also recommend that they get ADT with it? Since, at least in my opinion, but you'll correct me if I'm wrong, ADT and especially double hormone blockade, not just monotherapy. Would you not recommend? I'm trying to get your formula. Let's say I'm BCR, which I am. Let's say I want to go off-label, but I don't have the resources to go to you to prescribe something off-label. Would you not prescribe to me double hormone blockade informed by the EMBARK study, in particular enzalutamide and Lupron plus your inhibitor? I guess that's a question.
So, I mean, if I could answer that in the very specific question, but I'll answer in the general way, which is really, I would say that these drugs, this pathway, this target, I think it's very interesting by itself. And again, we didn't talk about all this data, but there's overwhelming data that the MAOA is a target in preclinical models, but it's still a gulf, but some data showing it's a target in actual patients, at least suggesting it's a target in patients. But what is true about MAOA as a target and other targets is, I think there's a big advantage in oncology in treating multiple targets, hitting multiple targets, in that you can co-treat, with a combination treatment. I think you're alluding to like the EMBARK study, which was enzalutamide monotherapy, Lupron, or Lupron alone. So, or GnRH antagonist alone. So it would be useful to try to think about, I think, adding monoamine oxidase to other therapies. Yes, that would be my first thought if we were to really implement this agent or this strategy. But it's it's very much an open question, and testing the combinations, again, as a clinical trialist, as a researcher, as a scientist, and as a physician, it's hard to know when you add the combinations, which is one better than the other, or the combination better. So this is the problem, again, the very practical problem facing the field. So,
Alan, you're on mute.
thank you for the question.
Alan, you're on mute. Let me stop the recording, and if those who want to stay on, we can continue to chat.